So we're going to look at blood types or blood groups, what they are and why it's important to know your blood group before surgery, etc. And if you have a sample of blood and you let it settle out or centrifuge it, you'll get 3 layers. The top is plasma, a straw-coloured liquid, about 60% of the blood. Thin layer in between of white blood cells and platelets, and then the bottom, the most dense, the oxygen-carrying red blood cells. And we'll review at the link at the bottom. And close-up, magnified view of blood. On the left, it's a granulocyte, white blood corpuscle leukocytes, and there are 3,000 to 9,000 per millimetre cubed. Be 1,000 times more in a centimetre cube, size of a sugar cube. Red blood corpuscles, erythrocytes, 4 to 6 million in a millimetre cube. Blood platelets, which are thrombocytes for clotting of the blood. And surrounding all of that, the plasma, it's a fluid, 60% of the blood, mainly water with some protein, lipids, glucose and electrolytes. So how much blood do we have? Adult, about 5.5 litres, and about 10% of the volume of the human body. And pause to look at the other animals listed there. Humans have 4 main blood groups, A, B, and O, and cats have 3. Dogs have 13, 8 are red blood cell based ones, and horses have 7. Sometimes I think you'll see it done as 8. And the person who discovered it was Karl Landsteiner and defined them as ABO blood groups in 1901, although called it ABC to begin with, and the O came in later for the German owner without. And then we have the blood antigens on red blood cells, and the antigen precursor is the rhombus on the left-hand side, called substance H, sometimes called antigen H. And then you've got the precursor on all the types of blood, but you've got a different group added to A or to B, and you can see what those are below. And then for a blood transfusion, you have to have blood that won't clot, that doesn't have the antigen that causes the clotting. So blood type O is a universal donor, and AB is the universal receiver recipient. And here's a table which you can look at more closely with the question, who could receive that sachet of O positive blood? What blood groups are compatible with it? Pause to do that. There are also blood antibodies in plasma. On the top line you see the red blood cell type with the antigens on it, and the antibodies are in the plasma. Plasma is the serum with the antibodies in it, and here you'll see group O actually has anti-A, anti-B, and group AB has neither of those antibodies. And if you're having a transfusion of plasma, AB is now the universal donor and O is the universal recipient. And look again at that compatibility chart. And so we have blood group phenotype, which is how you can test, perceive the blood group here, and the genotype, which is DNA derived, 1 allele from mother, 1 allele from the father. You can pause to look at that one. And then we have a look at the distribution in the US of the different blood groups, and the one on the left is the whole average, and the one on the right shows the phenotypes by race or ethnicity. and pause to compare those. Then we can look at the blood groups by continent. These are just O+, A+, and B+, haven't got the negative blood groups in there, and pause to look at the differences, and can you think of any reasons why they're different at all? Difficult question, that one. And here's a little bit more detail, and it shows some of them here where you have indigenous original peoples of a continent, subcontinent, compared to what they are today with different migrations, etc. And then there's the rhesus factor. And so the antigens on the surface of the red blood cells are carbohydrate based. And the rhesus factor, again, it's on the surface of red blood cells, but it's a protein based unit. Rhesus positive red blood cells have the protein. Rhesus negative don't have the protein. And you can transfuse rhesus negative blood to both rhesus positive and rhesus negative, but you can only transfuse rhesus positive to rhesus positive carriers. And on the right, you see a mother who's rhesus negative, but a baby is rhesus positive. The rhesus negative allele here is the recessive, and so the father must be rhesus positive. And again, you can pause to compare the overall distribution of rhesus positive and called rhesus D for the main rhesus antigen, to those by race and ethnicity. And again, pause this one for a little while to see the differences. What are the antigen add-ons or to both the negative and the positive for phenotypes? And look at the rhesus factor by continent. Different countries are looked at there, and look particularly at the differences. And then we're going to look at a blood typing activity. You can read what the activity is there, the crime committed with Miss Hong's classroom. But we're going to have one blood sample from the crime scene and 4 suspects, and their different 4 blood samples, and all the samples need to be tested. And what you need to know, if you test blood A with antigen A, you'll see clumping, agglutination. You'll see clots. That'll happen with type AB, so you need to test with a different sample with antigen B to check that it isn't AB if you'd got a positive test with antigen A. So read that fairly carefully and to see what happens with type O blood. And the materials you need, the well plate with the dimples in, simulated blood, and simulated antigens, A, B, and the rhesus factor. And what you're going to do is half fill 3 dimples with one particular blood sample. Add antigen A to one dimple. Note what happens, any clumping. Add antigen B to a second dimple and the rhesus factor antigen to the 3rd blood sample. And complete the table of results at each blood sample, and then continue with all.
So that's when the blood, it starts to kind of become clumpy. So when you add the anti-A or anti-B, whatever reagent, If it doesn't clump, then that's negative. It just
Too little.
so
Okay.
anti-A,
1, 2, 3.
so the antibody reacts.
Okay. Now we'll do anti-B in one of these.
3 drops of anti-B.
Yep. One, 2, 3. Okay. This one looks like it will be promising. Okay.
And that one's already coagulating.
1, 2, 3. And these two seem to be.
Alright, okay, so you put separate ones in that time?
I put anti-B, anti-RH.
You started at a special one where all
Strange. Okay.
Let's go.
I think I was probably.
Okay, that does
No,
not mean
no, just do it again.
You can kind of tell by the start.
Is that orange?
What's in that bottle?
That's A.
Positive for
Look
A.
at the bottle. Great.
Positive and negative.
One more to do.
It's like a very federal type case. What do you mean? Anti-B, which means that it's AB. It's AB. AB minus.
4.
You can do all the cleaning up.
This is 4.
It has to be you.
Negative.
It's negative, positive, positive.
Of the 5 blood samples. And there's the table of results which you can put on your results. And if there's clumping, it means there was a reaction to that antigen. So antigen A clumping means that that antibody A is present in the sample of the red blood cells. And you can go through them and you'll see that in this particular case, only one of the suspects fits the crime scene. Still doesn't mean they were guilty though, does it? And the simulated blood was milk with food colouring. The anti-AB or rhesus samples were either water or vinegar. And if a positive sample test and clumping results, it would be vinegar that's been added. So the contents of the labelled antigen bottles for each of the 4 the suspects are different, and indeed for the crime scene.
Sure.
It's real blood. Why would you want to drink it, right?
And a little review here, looking at codominance, multiple alleles, and the phenotype and the genotype as well. And a question: what are the possible blood types of the children if mother is type AB, father is type B? Use Punnett squares to work that out and pause until you get a response. And one Punnett square shows homozygous alleles on the left, and on the right here with heterozygous alleles. And pause until you have a response. So you can check your response with that, go through that fairly quickly. Question 2. 3 babies are born in a hospital but switched from their cots at birth, and which baby belongs to which pair of parents? And so does 1, 2 or 3 belong to X, Y or Z? And so work out what are the possibilities and which baby can only belong to one of those sets of parents. And so you've got a response there that you can look at closely. And so some links here that might be useful: online Punnett square calculator, online simulation, online blood compatibility. And
coagulating, then that means that it's positive.
Okay.
Yeah, that means it has the
So, this is A.
very many thanks to the student in Miss Hong's biology class and Washington International School Team Science.